I'm excited to be moderating this panel. I'm Claire Aldridge, and I've had the good fortune for the last over 25 years now to get to participate in the biotech ecosystem, really translating scientific discovery into the marketplace. And I've gotten to do that as a venture capitalist at startups, as well as at academic medical centers. So the thing I'm most passionate about is how do we take those amazing discoveries that our foundational scientists make and turn them into products that change patients' lives. And I'm really excited to get to talk to these folks about gene therapy. I think everybody in this room understands the potential of gene therapy and how we have the ability now with some of these tools to really change the trajectory of patients' lives with kind of one-time dosing and really disease-modifying technologies. And so we're going to talk a lot about that, where the field is going. But first, I want to ask our panel to do an introduction of themselves, and also recognizing that we are the panel in between your ability to ask all the questions you haven't been able to ask, and more importantly, get to the cocktail hour. So with that, Michael.
So I'm Michael. I lead the technical development and operations group at BridgeBio. So that's, as Brooke mentioned, our analytical development group, our process development group, but also the team that manages and supports all of our clinical manufacturing and our CDMO partners.
Paul?
I just forgot. I think it's not.
Okay, is it on? Yeah, okay, perfect. So Paul Herzik, Chief Technology Officer at Solid Biosciences. So my responsibilities are manufacturing, process development, basically anything around supplying AAV from mice to humans.
Fred?
Fred Porter, Chief Technical Officer at Taysha Gene Therapies. I saw you all before.
It's good to be here. I'm Sebastián Aguirre, co-founder and head of platform development at Carbon Biosciences. Carbon Biosciences is probably the only company that is not purely AAV, but we still use the AAV genome, and we are packaging in parvovirus capsids, so it's even more challenging. So I'm happy to discuss more.
Beth Ann?
Hi, I'm Beth Ann Childress, head of our GMP quality group at AskBio.
Wonderful. Thank you all for being here, and certainly thanks to Forum and all of our partners for helping us put this on. The first question, I think everybody's alluded to this, that gene therapy has had its ups and downs, and it certainly has been in a little bit of a trough, gene therapy winter for the last couple of years after some really exciting things. But I think that, we all think the potential of the modality is there. So what I'd like to ask the panel first is to talk a little bit about what is it that you're most excited about with gene therapy? What do you think is going to happen from an innovation perspective in the next 1 to 5 years that could really kind of bring us out of that trough and really start to accelerate the approval of these things for more prevalent diseases. And I'm just going to call on people and keep it lively. So Sebastian, would you start?
Sure. So what I saw today is really invigorating. Also seeing the progress in the field, especially in the last couple of decades. So it works. We are seeing transformational work, really curing people, which decades ago would be biblical, really, to see this effect. So and that's invigorating, and so we really need to work hard in solving the bottlenecks, I think, and this is a great forum really to discuss these bottlenecks and the challenges. But I'm very optimistic.
So is there a particular bottleneck you think we'll be able to target first?
Okay.
Sorry.
Just, manufacturing, again, safety, efficacy. So, yeah.
All of them. All right, Beth Ann?
We can double click, of course, in each of these and spend hours discussing about
Well, we could, and then nobody would ever get to the bar, and they would all be very angry with us.
I think Sebastian covered some really important pieces that we'd probably all agree with. To me, some really exciting aspects is we have some great programs coming through the pipeline right now at multiple companies. You represent many of these. I think there's a real excitement about opportunities in larger patient populations for therapies that cardiovascular or oncology or infectious disease, and so
neurology.
Yes, I mean these are amazing. I think the excitement with that is that as we are able to impact these populations, then it's going to normalize gene therapy, hopefully in the patient populations, but also within the physician landscape. And so we'll get a little bit more foothold into like the public awareness. So I think that there's a little bit of some momentum once we start getting more progress in there.
I think access, right? Like, how do we get these to community physicians, right?
Yeah.
People who aren't seen by academic medical centers.
Yeah.
Fred?
Yeah, let's see if this is on, I could just yell too. What I'm excited about is how we've been moving forward, like with regulators, and I think there's been a lot of, I think collaborative work going on. I think it's important to mention the START program where cell and gene therapies have been named as an area of focus, and I think, what I've seen recently is when we come to regulators with programs, they're more amenable to work with us. We're closer to understanding each other and what the expectations are. So that's what I'm excited about. And I think our understanding of how to move these rare disease programs forward is clearer all the time. Just a few years ago, you would start a clinical discussion with the agency around having a placebo double-blinded trial. I think there's a real awareness now that doesn't work for many disease indications, and that's really important.
And I think
because-
is even, I'll be bold to say, unethical.
Yeah, but indeed, I think that conversation has changed, and I think that's a huge sea change in the way we think about these trials, because we are all focused on design and delivering vector, but really at the end of the day, we have to show efficacy in fairly small populations for most of these diseases.
And I think we can't just pattern it after what they did for small molecules, right? This is a very different thing. Paul?
Yeah, I think coming back to what you mentioned about the winter, right, and how we've seen, I think the biggest change that we've seen over the last couple of years is, tied to investment in this industry and how that has gone down, but it's still there for us to all grab. And really where that shift has changed is, when I first started in AAV, you could get $200 million for an idea. Now you get $200 million when you can prove that it works.
When you have phase 1-2 data, right? Yeah.
Right, but it's still there and the money flows, but it's the expectations, and with the high interest rates, it's all been kind of devalued for the point where investors want to de-risk their investments as much as possible.
And I think in some ways, which I'd love your thoughts on this, Paul. Like we as a modality were penalized by COVID, right? Because it was the hot modality when everybody decided that science was cool, right? We mRNA vaccined our way out of this, and so then all of these generalists dumped their money in.
I've been saying since COVID, you've seen money flow into RNA and out of AAV for that exact reason, right? And it started almost in that time frame.
Yeah, Michael?
Yeah, I'll just yell too, like Fred. But
Happy to share.
and
You
just-
can moderate.
What was the question? There's been a few.
What are you most excited about? What do you think is going to happen over the next, let's say, 1 to 5 years, knowing that science never goes as quickly as you think it would, that will change the trajectory of gene therapies and the approval, the development?
Yeah, so I'll touch a little bit on, I think, what Robin mentioned in her talk. I think there's been, 7 AAV-based gene therapy approvals. Is that roughly right? And I think 3 of those are considered blockbusters, and I think those are pretty good.
That's right. Yeah,
and I think
no small
investors-
molecules
yeah,
can say that.
Investors, I imagine, pay attention to that stuff. And like you said, Paul, maybe it's, it changes when you can actually raise money, but I still think those opportunities are absolutely there.
Well, and the reduction in risk in going through, how many small molecules fail along that path versus gene therapies. I think those are all really valid points, and I think we're, as they say, preaching to the choir. We all agree, but it is nice to kind of hear the breadth of those answers. The next thing I'd like us to talk about is that regulatory environment. Fred touched on that a little bit. Peter Marks has come out and said this is an area he would really like to be more of a partner with the companies. And one of the things I think about innovation is kind of the space I like to play in, and I always try to explain to people, like, if it's really innovation, there's not a playbook, right? It has to be, you know, kind of bespoke every time. And this is really innovation. So there's not a regulatory playbook the way you might have with a small molecule. So, you know, we've got that side. Then we also have, you know, kind of what is happening with the new administration as far as cuts and things like that. Don't want to get too political, but just kind of what are you seeing in that regulatory environment, kind of expanding on what Fred talked about. You know, do you think this is going to get easier or harder? And does anybody want to go first? I see Beth Ann. You
Well,
like?
I think Fred raised a great point. Our regulatory agencies are actually, they've helped us quite a bit in the last few years between the Bespoke Consortium, Interact, the RMAT designation, the gene therapy pilot program. So they've actually done a lot to help us in that space. But right now, I think, are seeing some potential challenges with the administration changes. We've got some capping on indirect costs. We've got delays and cancellations of NIH meetings. I think there's some grant freezing. So there's a little bit of instability. Hopefully it's short term, but I think that there is potentially a trickle down effect from that, because any delays or funding concerns or inhibitions to really talking about important research data does trickle down into our development space and into our clinical space and commercial space. It's hard to kind of make up that time and that funding. So I'm curious what the others think on that too, but that's, you know, I'm thinking maybe a little bit concerning right now.
Yeah, I mean, I took a note, that's why I have my notebook. But, so I would say in general, as long as Peter Martz and Culver Dunn are around, I'll feel pretty good.
They
But-
have been good partners.
Yeah, I think so, but and before I put this down, I'll make sure it's off. But I was reading an article the other day, and there's something that made me laugh, and then I kind of got some of these questions, and so I dog-eared this page, and it was about, you know, that HHS had some sort of like roundtable discussion on, you know, cell therapies, specifically stem cells. But one of the quotes was that that the FDA is going to stop their aggressive oppression of psychedelics and stem cells. And so I read that and I kind of I thought it was funny, but I kind of wrote it down. And here I think, like, look, I don't know. I mean, there's plenty of reason to be concerned. But man, I think to myself, well, I mean, if they're cool with like LSD and psilocybin, then
Gene
probably,
therapy's taking a spin all of a sudden.
It's reasonable to think, you know,
Gene therapy
they'll be
with a
okay
twist.
with, you know, gene therapy.
Yeah, right. You're going to be like normal, right?
Yeah,
It's not
you know,
even
yeah, so-
barely cutting edge anymore. Yeah. No, I think that is just as long as they're using evidence-based.
That's right.
It's
Now, I
on
get
them.
what I can't, yes, exactly.
Fred, did you have something to add?
Yeah, I mean, I think we all have a similar concern. I think, at least to date, haven't seen any of those concerns come to fruition. So that's good. So far, so good, I would say. You know, I think let's just give a nod to ex-US regulators too. EU just came out with new cell and gene therapy guidance that's really clear. That's been really helpful. You know, they have great avenues for engaging with regulators there as well. We've had great conversations with Health Canada in the UK. So it's more than one regulatory body that you can have great engagement with. And so I'm encouraged by all the advances so far.
Well, I guess, and this is, I reserve the right to go off script. That just reminded me of, the conversations that I've been reading about over the last few weeks about the U.S. talking about, putting pressure on Novo and Lilly with the GLP-1 drugs, right? And so, I think that that's completely not appropriate for this audience, but that does lead me to your comment about the ex-US. Are there any things that you're seeing about the United States trying to put any pressure on those, advanced therapeutics, gene therapies, cell therapies that have a much higher price tag, but I think from a health economics perspective, absolutely justify that price tag? I would just kind of love any thoughts you might have on that.
I think for this audience, all of our products are too early to say.
All right.
Too soon.
Okay, all right, fair point. So, but kind of going on that, you mentioned, Fred, about the small patient populations. you know, again, this is a modality I think we know works, right? If we can get the right payload, we can get the right vector to take that in, we know that we can change these patients' lives. But then it does become a, how do you even run a trial when there's 12 patients in the world? Or, you know, something like that. So I would love to know your thoughts, kind of how do you balance that, you know, the potential of these therapies with the risk of moving them forward and the size of the patient populations?
Something that we were discussing today is how we apply innovation and new tools to the early stages of this development. But I can't wait to see also how AI and all these new modalities also help in the later stages, accelerating clinical trials. I was checking recently one of the new companies that is proposing this, and they say instead of, typically you need 50 people to write and prepare a clinical trial, now we can do it with 3. And imagine the impact that could have in developing all these drugs and extended accessibility for these small groups of patients. So I'm optimistic that it can be great.
Paul?
I think one of the things that the last session Adam talked about, we talked about scale up. I think thinking about the end in mind from the beginning is very critical. Like one of the things that some of us put in place in a previous life was we were dosing mice with what we intended to be our commercial manufacturing process. Mm-hmm. So there is no comparability, there is no changes. It's just what you're submitting at the very beginning that's going into your mouse, is going into your tox, is going into your humans, and you are set from the beginning. That simplification of it allows you to go through the regulatory pathway much easier if you have the end in mind from the very beginning. And if you can establish systems to do that, as you are thinking about a product, it allows you to go much faster through the regulatory process and through the phases much faster.
So how much of a risk would you say with that, of that potentially failing in mice?
Well,
Right.
so I mean, you can make it all depends on how you do your
Right.
mouse studies, and this was not my area of expertise. He's sitting out there, right? But, you can do a small prep to get proof of concept in wild-type mice, but then when you're going to do your IND-enabling studies, your dose finding, your efficacy, you take your tox batch, which should be at your commercial scale, and you filter some material to run those studies in parallel, and your IND package is pretty.
Yeah.
I think I'd even add to that. I think we don't talk too much about the animal models sometimes, and I think thoughtful and strategic animal models really helps us identify which programs we want to progress sooner. It gives us the right data at the right time to make thoughtful and robust decisions on programs. So I think that's a great point about the animal models.
Are you seeing any appetite from the regulatory agencies on acceptance of like patient cell data?
I haven't personally. I know there are particular capsids where that might be part of the conversation, right? So where you don't have a good animal model, you might need patient cells or human cells as the surrogate for some of the animal pharmacology.
I'm just thinking about, one particular indication, right, where the animal model doesn't have a phenotype, right? And so how do you can certainly show safety, but it's a little harder to show efficacy. And so, how do you do that? How does the agency partner with you to, kind of answer that question?
Yeah, I think on the converse, say you have a capsid that's very tissue specific to human cells. How do you put together that package? And I think that might be part of the equation. Those conversations are ongoing.
Yeah.
In the absence of phenotype, and when you say phenotype, you mean, I guess, evidence of disease.
Evidence of
correct?
disease, right? Yeah.
But I guess, right, at some point we all have to count on, depending on the disease, depending on the patient population, you've got to count on these regulatory agencies understanding the challenges that face these programs. And you got to count on them, not only understanding, but giving certain amount of latitude, when it's appropriate to give that latitude.
Given the risk, right?
Yeah, but we do a lot of studies where, I would call, certain aspects of a biodistribution study a phenotype. I can see, RNA get into cells that we intend to target. I can see protein being made in those cells, and I certainly think that's a phenotypic change in an animal model. And so I do think you just have to be prepared to make a creative argument and hope that a regulatory agency is open to
Right.
hearing, your argument in the absence of the ideal.
Yeah, well, and again, I think this is a new modality. It's not as straightforward. And I made a note about the collaborative cross. I think that's how do you more accurately reflect in your development kind of the genetic diversity of the population, so that you can potentially see some, signals that you might not otherwise see until you went into a large number of patients. Okay, so for the final question before we open it up to folks, would just like to ask you guys, what do you think is the biggest challenge for the industry to target in the short term in order to capture the potential of this modality?
Funding?
Funding? Okay, all right, that's,
Certainly, I think we touch on several here: manufacturability, safety. Another aspect is getting to the full population. Many of the AAVs are very circulating in the population, so everyone has antibodies against that. So that's a big issue, I think.
I continue day to day, I guess, as I'm thinking about this stuff. I mean, I always return to starting materials. So it's the vectors that we use to make these vectors, so like our plasmids, as we improve those starting materials, as we improve the cell lines that we're using to manufacture these vectors, I think the change there, and going back to your first question about, what are you excited about over the next year or over the next 5 years, is really advances there, because I really think all of this is going to change dramatically, even as iterative improvements in those starting materials are made. When you talk about productivity levels, when you talk about, like so many talks today about, product quality, and really starting with, hey, I need to design these starting materials because I care about these certain product quality attributes.
well, because that also becomes exponential
Yes,
as you
and
go through the
then
path.
it's going to change a lot, I think, in terms of being able to, run trials more quickly, run trials less expensively, reduce the cost of manufacturing. So that's kind of what I think about a lot if I'm brushing my teeth, washing my hair.
I mean, I just want to add to that, right? I mean, there's been good and bad things about the lack of investment. We have traditionally seen in the last year and a half, prices come down for manufacturing a batch at CDMOs, right? The demand is down, the supply is up. Basic economics means prices are coming down.
Right, you don't have to put $1 million up front to get your slot in 18 months.
And when I first started using CDMOs, whatever it was, 6-7 years ago, it was 12 to 18 months to get a slot.
Yeah,
Now you can get slots on a routine basis. This is allowing us to drive down the cost of getting to a clinical trial. Right? So you can get more shots on goal, you can get more products for less investment.
yeah, you don't have your burn while you're just waiting for your slot.
Exactly.
I have a different perspective. I think that I think we can address all these manufacturing issues, and we can, and we do. I think the area that's been more difficult is aligning with the FDA on what are approvable endpoints. How do we use natural history? Can we use biomarkers as surrogates? Those are the things that seem to be, whether we deliver, when we deliver a product and get it ready for the clinic, it's really the decision-making around that and alignment on that and how rapidly we can do that. That's really going to make the difference between approvals and getting these products to patients. Some of them are not, lethal diseases like Zolgensma, where you can use life or death as a measure. They're often more phenotypic, behavioral. And you heard from Jude about, look at those wonderful videos. Well, that enough is not. That is not.
It's not an
sufficient with the
approvable
agency.
endpoint, yeah.
So how do we quantify that? And it's been really difficult in the field. I think we're all working together, but it's not that simple.
Yeah.
And I think that's where I would double down, is to have those conversations. And every disease is different, which makes it more challenging. But
That I think
that's
is,
the opportunity in my view.
yeah, it's not like there's one endpoint you can do for all gene therapy.
No.
You know, it's gonna have to be, and that requires the agency having an understanding of the disease,
And they
not
also
just the
like
modality.
To retreat to well-known endpoints that they're familiar with. Some of these diseases just don't lend themselves.
Well, they've only recently been known to be a separate disease and not just lumped into pediatric epilepsy.
So
Mhm.
that's my thought.
Beth Ann, did you have something?
I think all of these points are great and extremely valid. Maybe if I was going to add a few challenges, I think supply chain challenges with getting the product to the patients and where they're at and where they can have follow-up. I think in addition, the analytical strategy is always really critical. And we are given a lot of help by the regulatory agencies, I think, with these programs, but they also expect more from us earlier now. So I think those are some additional challenges we'll have to continue to work on.
All right, I would like to open it up to questions from the audience. Yeah,
I can be loud.
I can also repeat.
So, I mean, very educational, learning about all the NGS work that's gone to, you know, basically characterize the different fragments. But curious, you know, do you believe there's going to be a change where you have to characterize what those fragments could do within your process, right? Like off-target effects. We're not talking about, I mean, maybe I'm just haven't read more about it, but the off-target effects of those different fragments.
Well,
do you
right,
think
if a
that's-
truncated protein doesn't have a regulatory region, for example.
Yeah, but is that going to be like a profile that's going to be part of the safety assessment for an AV therapy?
Yeah.
I think, well, that was,
It was in your presentation.
yeah, I think that was part of Brooks' talk, you know, is how do we take a closer look at this specific population that's often just considered a product-related impurity. When actually, you know, we want to be open to the idea of defining it as a product variant that still has some measure of therapeutic potential. You know, and so Brooke showed, you know, a couple of slides with data from a RT-ddPCR assay where we can look at mRNA expression, and those intermediates absolutely, you know, express message. You know, we also have from a different program, you know, a functional assay where we can see enzymatic activity after, you know, transducing cells with these intermediate species. But again, I think as Brooke mentioned, you know, we want to take it a step further and go into animals, because just because it's efficacious doesn't mean it has the same safety profile as, say, your foals. And so I think those are the types of studies that we want to do. You know, I think we just feel it's our responsibility as the sponsor to really understand what the hell it is that we're putting into these patients. And so, yeah, that's one approach at least that we're taking to understand that and use NGS to help supplement all of that data.
Ben?
Just with regards to, again, these diseases that have a pretty bad endpoint, someone's going to die. Do you think there might be a regulatory pathway in the future where in order to get these therapies through faster, there might be some sort of compassionate use lane that could be applied?
I think we're actually seeing a little bit of evidence of that with the RMAT. I think they do want to, I think the agency does want to have grace. They recognize this, and certainly the rare disease advocacy groups are very vocal in pushing that. I'm not sure how much the compassionate use has been used for gene therapies, though. But
We know of at least 2 or 3 different
Yeah.
occasions where that regulatory pathway has existed for a very long period of time, right? It's not used frequently in gene therapy, but we know, right? Barry Byrne and Guangping did a trial at one point, right, in Canavan disease, and there's been a couple others. A lot of parents are advocating for their own children, trying to move, you know, AAV programs for their particular child through you know, CDMOs and things of that nature. So there is a pathway. It's just not. It's typically less compassionate use and more, you know, principal investigator-led trials from an academic standpoint.
I think as that patient population is greater too, that compassionate use is going to be less likely to be able to be used.
And then to follow up on that, do you think this could be something that does get used as viable clinical data to look at least? Or is, again, it's just like, this is a one and done thing, you can't use it for anything else?
In our previous life, we would have loved to have used it. We couldn't get access to the data, right? Because it was behind, you know, an investigator study, but, you know, I think and, you know, how we would manufacture versus how it was originally manufactured, we're all different, right? So, I think it depends. Once again, if you have the end in mind and you're doing that, yes, you could probably get away with it. But if you're doing it as a one-off, as a small batch in an academic lab, from a manufacturing perspective, it's gonna be hard. It's gonna be, once again, coming back to that regulatory hurdle of you know, compatibility or comparability, it's gonna be a challenge to put that in.
Yeah, it's just, I could respond to that too. I think it might be helpful for clinical de-risking. You know, I don't think it's a springboard to faster approval. So I think that's probably where it might be.
So there are examples, if the patient's in a very bad condition, you could run into it within a week. So they would allow you to, so let's say this patient is dying
Yep.
and there's no treatment. You can, there are examples in the MPS, lysosomal storage disease field.
Yeah, very much
And,
so.
yeah, and also I want to give you guys some more positive regulatory insights. So very recently, at the end of January and the beginning of February, there's a World Symposium on lysosomal storage, lysosomal diseases. Peter Marks gave a talk. It's a very updated talk. So now for rare diseases, if you do AAV, gene replacement therapy, and they have a program established called platform gene therapy. So you're allowed to, if you have several diseases using exactly vector, except you swap the transgene. So you are allowed to do just one IND for all those diseases.
The caveat to that program, though, is you have to have one through BLA.
Yes. So you don't have to have all, you know, let's say we do lysosomal storage diseases. We have 4 or 5 targets. So you could only, you only need to get one IND approval for those trials. Another one is about the biomarkers. So apparently it's almost to the point they will allow you to do the specific biomarkers for each disease target. If you have the positive biomarker changes, and you don't have to have long-term functional impact data, so they will let you to go. You know, there's another one, a very good program. I can't remember the name. If you get on FDA website, you will find it. It's all designed for help rare disease gene therapy to go through.
Will, did you have a question?
It's kind of long-winded. So
Oh,
I'll say it.
all right. Other questions?
Oh, adding on to Michael's point about what we are putting to the patients, I just wanted to add one more point. While we focus on vector development, improving designs, etc., improving integrity, one of the things that we probably not much appreciate is how much we dose patients, right? Right? I mean, we are still stuck to the age-old method of qPCR. We know, genome fragmentation happens. Is that the right method? How are we dosing patients? I think, adding on again, Michael's point, I think as an industry, we need to appreciate what genome integrity means, how we redefine, how we dose patients, how we measure efficacy, and advanced methods to, improve how potency is measured in vitro and in vivo as well. I think that's where I believe industry has to come together to redefine, how we basically do, do clinical trials and efficacy studies and how we measure dosing.
I think it's even worse than that, right? The paradigm in most IV AAV-based dosing is VG per kg, right? You might not be targeting, we are massively overdosing patients for trying to get a viral particle into a particular cell because we're trying to overcome the liver sink, right, at the end of the day. And we are overloading immune systems, right? And let's be honest, we're wasting virus not going to the places we want it to go to, and once again, it boils down to delivery at the end of the day.
Think of how many more doses you could get out of that. Last question, Adam?
My question was, are being placed in front of them. And do they, I mean, is it really, do they need to have learnings? Right?
And your
In
role
order
as a teacher
to-
in that.
I mean, they're acting as the judge, right, for a lot of these things, and in that position, it suggests that you have the right knowledge base to make those judgments.
I will say that we have definitely seen, I won't comment on what they know and don't know, right? But what I will say is,
In general.
we have seen in the last 12 to 18 months, coming back to one of the original questions, we've seen far more flexibility from the agency to things where they used to be very dug in on. You must have a potency assay that is mechanism of action related. And with their potency assurance guidance document that came out in December of 22, I think it was, you can now take a compilation of assays and build a portfolio around that and not have to actually deliver a true mechanism of action potency assay, which is a gigantic change in perception, because that was cut and pasted on every IND that I have ever seen, right, from an AAV perspective. And they have definitely, that document has opened up a lot of pathways from a CMC analytics perspective.
I think
Yeah.
I might, oh, sorry.
Go ahead.
I might answer a little bit more directly. I think each of us probably has a little bit different experience based on our reviewers and who's supporting our programs. But I think for me, the big thing is there's a huge difference versus 10 years ago or 8 years ago. So they do, at least in my experience, which may be different, they seem to be very knowledgeable, much more knowledgeable than they were in the past. So there's been a concerted effort within the agency, both FDA and EMA and MHRA, et cetera, to really educate their reviewers. And I think I see that coming through their questions, their responses when there's meetings. So, yeah.
And what I've been told, I've never seen, but I've been told that Cedar and Seaver have labs where they're actually doing work. And they're, so they are learning and they're educating themselves. I've never been to these labs, I can't, verify their
I've seen a
existence.
couple at a couple meetings.
Okay.
I've seen a couple posters and stuff.
All right. So there's that, but then also I think what keeps me up at night a bit is, well, they're also learning through every single interaction they have with companies like us, and the questions that they ask, they get different answers, probably, or sometimes similar or overlapping answers from all these different biotech and pharmaceutical companies.
So they get a breadth.
Yes, and
They always know more than you do.
expectations, the expectations are gonna be set, and what I'm worried, like, and what my team, and what we talk about is, well, we wanna be part of the team that's helping to set those expectations, and not ones trying to scramble to meet those expectations when we get questions we don't, or we haven't thought of, but so far, we're treading water and we're staying there, but, yeah, it is something that I do think, I wanna give the FDA credit that they are learning, and yeah, you can tell just by the questions you get versus an IND submission last year versus in 2019.
It's very different.
Can I add on to the answer to that question? We at the Alliance for Regenerative Medicine are doing some of the training of the FDA. They've come to us.
Stop that.
They've come to us and said that we want to learn about some of these new technologies we've never seen before. So our members have partnered with us to bring in usually academics, so we're not pushing anybody's particular product. But we've done 4 trainings so far. We've got a few more lined up this year. So they've really worked with industry kind of from where we sit a lot to learn this stuff.
That's a great perspective.
Well, I think the object of this panel, which I think has been overwhelmingly achieved, is to now take it out to the networking reception and continue this conversation with all of our experts.